In sub-Saharan Africa, part of the economy is driven by cattle, and cattle are one of the targets of the African trypanosome. And so this has a huge economic impact on that area, maybe even as much or maybe even more than just the impact of human disease. A lot of people have heard of African sleeping sickness, where the parasite resides in the midgut of a tsetse fly. When it bites a host, the parasite can get transmitted from the bite to the human. Once it's in the brain, it's always lethal. The current treatment for late-stage trypanosomiasis is based on an arsenic compound. Up to 5% of the people who get the treatment die from the treatment. The parasite uses glucose from the host to live. People have known this for a while, but no one has really targeted blocking glucose as a real avenue to treat disease.
So the gist of the project is we have a sensor in our cells. They change color if there's glucose inside the cell. Using that as a readout, we identify drug targets that kill the trypanosome.
Because the current treatment is so poor, it made sense that if we could kind of monitor glucose, we could look for molecules which would block glucose uptake.
Trypanosomes are very fast moving, so in order to take images of them, you need a very fast microscope. And so I was able to build and construct a custom microscope for our lab that allows us to image the trypanosomes, which no one else is able to do with traditional microscopy.
I was a graduate student once. You kind of know that you're done with a PhD when you're telling your boss about how things work. I'm happy when things work, when graduate students are able to make things work. I like to see them mature as scientists.
We have a pretty unique lab. We call ourselves analytical biochemists. We answer our questions with very unique methods.
It's still a challenge to figure out what's really going on in the parasite. Compared to lots of other things, there's not really that much known about it. I would say there's at least a decade, maybe a whole career's worth of work. There's so many unanswered questions. What can we do together?